I am Masayuki Saijo from the Department of Virology, 1 National Institute of Infectious Diseases. It is indeed a great honor for me to be given this opportunity to speak at Fukuoka One Health International Forum. The title of my presentation is Development of Therapeutic and Preventive Measures Against Severe Fever with Thrombocytopenia Syndrome, SFTS. With regards to conflict of interest, I have no COI to disclose in relation to the presentation today. These are the topics I'm going to cover today. First, I'll talk about epidemic status, new issues and challenges, in particular the transmission of SFTS virus to cats and dogs, which lead to the infection of owners and medical practitioners. These are some cases that have actually been reported. I'll talk about the development of treatment and the vaccine development. I'd like to share with you the most recent results at our lab. Severe fever with thrombocytopenia syndrome was first reported in the paper by the Chinese researchers, which was carried by New England Journal in April 2011. This is the paper with the title of Fever with Thrombocytopenia Associated with the Novel Bunya Virus in China. According to this paper, disease with fever, gastrointestinal symptoms, and hemorrhage and disturbance of consciousness in severe cases was reported in central China, in Hubei, Hunan, and Shandong provinces. As a prospective study, Chinese researchers isolated virus from the patient's blood. And from the blood samples, novel Bunya virus was isolated. Because we know that the pathogen is Bunya virus, and experts can easily guess that these viruses were mediated by mosquitoes and ticks. So the researchers studied the ticks and mosquitoes in the areas where the patients live, and they showed that Haemophilus longicornis carried the virus. According to this paper, the fatality is 15%, which is very high. About 1.5 years after this paper was published, in November 2012, a patient living in Yamaguchi without a history of travel overseas developed fever, vomiting, and melena. At first, the patient was observed on outpatient basis, but because the symptoms didn't resolve, on day 4 after the initial visit, she was visiting hospital, and the blood test showed a leukocyte count of 400 and platelet count of 8.9. Prominent decline was seen. This 400 of leukocyte count is 1/10 of the lower limit of normal. So because of this, hematological malignant tumor was suspected. So this patient was brought to Yamaguchi Prefectural Grand Medical Center on emergency basis. The bone marrow testing excluded hematological tumor, but in the bone marrow of this patient, macrophage, which is responsible for the immune system, was abnormally activated, and phagocytosis of the bone marrow was found. This is called hematophagocytic syndrome. A chest X-ray did not show any abnormality, but right axillary lymphadenopathy was seen, as you can see here. Other than that, there was no other abnormal findings in the image. This is the clinical course of the patient. After the onset of the disease, she was observed on outpatient basis for 4 days, and she was then carried to Yamaguchi Prefecture Gland Medical Center. Unfortunately, on day 3 upon admission, she died. Because the cause of death was unknown, she was pathologically autopsied. Right before her death, she had melena, and her blood pressure was unstable, so there were signs of multi-organ failure. There was virus isolation test on her blood samples, and from her blood sample, virus, as you can see here, was found. This is a round virus with 170 nanometers of diameter. From this morphology, this is the virus we can say belongs to genus Bunia virus. And viral test and gene sequencing led to the identification of the fact that this virus was the causative pathogen of SFTS found by Chinese researchers in 2011. So this SFTS was discovered for the first time in China, and 1.5 years later, the same disease was found in Japan. The next photo shows the swollen right axillary lymph node of the patient. The diameter is about 3 centimeters, and you can see that there are some hemorrhagic findings on the surface. And her stomach and large intestines also showed a prominent bleeding. Pathological test to see whether SFTS virus was in the lymph node was conducted, which used antibody against SFTSB nucleoprotein. So we used immunohistochemical test, and shown in brown is the SFTS virus antigen, and the swollen lymph nodes showed the existence of viral antigen. When this viral antigen positive cell is seen with the magnification, you can see a structure similar to large lymphocyte-like cell. According to a recent study, we found that this is the juvenile B lymphocyte. These images are from different patients. This is a patient diagnosed with SFTS, and the patient was brought to emergency outpatient clinic for vomiting blood, and emergency endoscopic test was carried out. This was by Dr. Shozo Kanayuki of Dohi Hospital in Hiroshima Prefecture. Emergency endoscopic test showed ulcerative lesions in the stomach, and from this lesion you can see the wheezing of blood. Thus, SFTS viral infection from the biological point of view can be classified as viral hemorrhagic fever. This disease was found in epidemic status for the first time in China, and there was an epidemic 1.5 years later in Japan. And at the same time, there was an epidemic in Korea that was confirmed. 2 years ago, SFTS viral infection was found. There were 2 patients in Vietnam, and last year there was a case in Taiwan. Although on informal basis, in Thailand, SFTS viral infection was reported. So, first, it was considered that the epidemic regions was in East Asia, mostly in China, Korea, and in Japan. But in reality, there was a wider distribution of the disease, more wider than we thought, in a geographic manner. Now let's see how these SFTS viruses exist in nature. In nature, a tick carries this virus. A tick with SFTS virus bite animals like raccoons and deer, and the mammals are infected. And among the mammals, virus replicates, and when the virus proliferates, non-viral infected tick would bite the animal and get infected, and ticks infected with the virus will lay eggs which also carry the virus. So in the natural scene, you have the cycle among ticks and cycle between ticks and mammals. And humans get infected by ticks carrying the virus. Sometimes, more recently, cats and dogs bitten by the tick develop SFTS-like symptoms, leading to fatal cases. So patients or infected cats and dogs can lead to the transmission of the disease to other humans. In Japan, ticks carry SFTS virus. There are 2 types of ticks. One is Hemaphysalis longicornis, other is Amblyoma testudinarium. And for the first time in November 2012, SFTS was reported in Japan, and later, based on the Infectious Disease Act, SFTS is classified as Class 4 infectious diseases, meaning diagnosing doctor needs to report the case to the authorities. According to the epidemiological study, there were 40 cases reported in 2013. In 2014, 15, and 16, about 60 cases were reported in each year. And in 2017 and onwards, every year we had about 80 to as many as 100 cases. SFTS is often seen in early summer. In summer, there's a decline, but then there's another increase in autumn, so we have 2 peaks in 1 year. So far, more than 500 cases were reported. Most of these cases were from western part of Japan. In particular, there were reports from Kagoshima, Miyazaki, Ehime, Okayama. So there were reports from Kyushu, Shikoku, and Chugoku, and Kii Peninsula. The northern limit of case report came from Ishikawa Prefecture. Earlier I mentioned about the SFTS viral infection of cat leading to a severe disease, and recently there was a case reported from Shizuoka in which a cat was infected with SFTS virus. So we can say that SFTS viral infection is gradually increasing in terms of the areas from where we see the reports. One of the major issues we faced recently is the fact that when cats are infected by SFTS virus or when dogs are infected, sometimes they develop severe diseases. These symptoms, clinical course, and signs are very similar to the SFTS viral infection of humans. Dr. Ken Maeda of the veterinary department of NIID showed that the SFTS cat fatality is 60%. And recently, we see cases where owner and veterinarians were infected by cat bite of infected animal, and some were fatal. Similarly, we also see SFTS infected dogs show SFTS-like symptoms, leading to the transmission to the owner, leading to the development of disease in the owner. So the owners and veterinarians and veterinary nurses are being infected. This is another case. This is a woman in her 50s living in western part of Japan. She was bitten by a cat who got infected, and she had fever, leukocytopenia, thrombocytopenia, and liver enzyme value increased, and she died on day 10 after the onset of the disease, and she had autopsied. The bone marrow testing showed the phagocytosis of the macrophage. So hematophagocytic syndrome was seen. If you look at the swollen lymph node, it showed the necrotizing lymphadenitis. So these were the findings very similar to the Japanese patients who died of SFTS. So we tested whether or not we can find SFTS virus in her organs in a pathological manner, and this is the result. SFTS viral antigens were found abundantly in the lymph node. So she was bitten by a cat infected by SFTS virus, and she developed SFTS and died of SFTS. So this was confirmed. So there's a serious issue of infections transmitted by pets. There were reports that medical professionals were infected by the patient, and there's also a risk of medical professionals, including veterinarians and veterinary professionals, being infected by the pets. And now we are developing the treatment which I'd like to share with you. Dr. Yosuke Furuta of Fujifilm Toyama Chemical developed favipiravir in the 1990s for the treatment of influenza. It's a base derivative, and the trade name is Avigan. When this is incorporated in the virus-infected cell, sugar chain is attached, and this OH group is phosphorized, and triphosphate is formed through the enzyme in the cell, and RNA-dependent RNA polymerase, which is viral synthase, is inhibited. With such activity, this compound inhibits the viral proliferation. And this mechanism has been validated. And its antiviral activity is found not just with influenza virus, but its antiviral activity is widely found with others such as filovirus, including Ebola virus, and Bunyan virus, including SFTSV. So together with the researchers of Fujifilm Toyama Chemical, we studied the antiviral activities against SFTS virus. Avipiravir showed the in vitro viral proliferation inhibition in dose-dependent manner. If you look at the EC90, it was 22 micromolar. On the other hand, ribavirin, which also has antiviral activity, we also showed that the SFTS viral inhibition of ribavirin in a dose-dependent manner. Interferon knockout mice were infected with the SFTS virus. In 1 week, almost 100% of the mice died. Using this model, SFTSV virus infection prevention effect of favipiravir was studied. And right after that, favipiravir was intraperitoneally injected once daily for 5 days. You have the placebo group, in which 1 out of 10 recovered, and the survival was 10%, and from day 2 of inoculation, you see the body weight reduction. On the other hand, if you look at the low dose group of 60 mg per kilogram per day and 300 mg per kilogram per day, in these two groups, the survival was 100%, and in the low dose group, there was 5% body weight reduction, which was small. If you look at high concentration group or high dose group, there was no body weight reduction. So clearly, there is the preventive effect according to this study. And next, we look at the treatment effect on day 1, 2, 3, 4, and 5. The treatment started after the onset. If the treatment begins within the first 3 days, then 100% of the mice survived. Furthermore, if we start giving this on day 5 and onwards, 50% of the mice survived. So clearly, in these mice, there is a treatment effect. So, together with the support of Fujifilm Toyama Chemical, and also with the collaboration with many parties concerned, we also studied the treatment effect of favipiravir among SFTS patients, and clinical study was conducted to see the treatment effect. So let's look at the results and the side effects. These are the results. 23 SFTS patients were treated. Unfortunately, 4 died. So fatality is 17.4%. According to the epidemiological study in Japan, in detail, the actual fatality of SFTS in Japan is 27%. Another study reports 31%, so the average fatality is around 30%. And with the favipiravir therapy, we were able to improve the fatality by about 10%. Of course, this is not a randomized control study, so we cannot say scientifically that there's a treatment efficacy, but I think this result can give us some suggestions of treatment effect. Now, let me talk about the vaccine development. Smallpox has been already eradicated globally, so smallpox vaccine is not in use at the moment. But in Japan, the third generation highly attenuated smallpox vaccine of LC16MH is developed. This uses the vaccinia virus. This is a double-stranded virus with a large gene. This is highly attenuated, and the test shows that it has effect against smallpox. This schematic shows that LC60M8 has the highly attenuated effect, and for the control we have B5R substituting with EGFB gene, and as vaccine candidates only the gene that expresses glycoprotein is inserted, or only membrane protein gene is inserted. Another vaccine candidate has the both gene inserted. If you look at the inoculation to the cell to see the protein expressions, in the control no membrane protein nor nuclear protein was expressed. Those inserted with genes that only express membrane protein expressed membrane protein. Those inserted with genes that only express nuclear protein expressed nuclear protein. And those inserted with genes that express both proteins expressed both in the infected cells. And interferon alpha receptor knockout mice used to study the viral activity were immunized with these vaccine candidates twice with 2-week interval, and then they were challenged with SFTS virus 4 weeks after the first immunization. And we studied the effect of these vaccine candidates. These are the results. In the control group, in about 9 days after infection, 90% of mice died. When 3 candidates were inoculated, the mice survived 100%. There was no body weight reduction. So each one of these candidates shows immune induction, that they were effective as vaccine. However, this nuclear protein immunized group did show benefit, but this immune induction power of this candidate was less compared to the one that only expressed membrane protein, or the one that expressed both nuclear and membrane proteins. The results of this study were shown on this site. You can have a free access, so please have a look. So today I talked about SFTS. SFTS is a disease that should be classified as viral hemorrhagic fever. One of the major issues is the infection of medical practitioners, and owners can get infected by pets, and pets also get infected. So these are very serious issues that I talked about. I also talked about the development of treatment, particularly about the treatment effect of favipiravir. I also talked about the vaccine development study underway at our lab. There are many unresolved issues which we need to overcome. So we have to wait for the results of future studies. This is a paper carried on Nature. It is a comment. To avoid SFTS infection, you have to be careful not to be bitten by ticks. Of course, that's very important. But even if you educate people with that information, you cannot get rid of the entire risk of SFTS infection. Researchers, scientists, vaccine manufacturers, and government, we all need to gather together our efforts to develop treatment and develop a vaccine. And that should be the way that leads us to the prevention of this highly fatal disease. In my presentation today, I talked about the favipiravir treatment study. This was conducted by Dr. Yasukawa and others of the Department of Internal Medicine of Ehime University. Other studies were contributed by NIID, Department of Biology 1, Department of Infectious Pathology and Infectious Epidemiological Study, Toyama University, Okayama Medical University, Yamaguchi University, Yamaguchi Prefectural Grand Medical Center, and Fujifilm Toyama Chemical. And I'd like to express my appreciation to all these researchers. With this, I'd like to end my presentation.